Hello everyone, welcome back again. So this is your next one chapter, chapter number 6, molecular basis of inheritance for the your class only, and video number 33, part number 1. So in this part of video, first we take the introductory point and whatever present in this video, it is the part of your NCERT, lines of your NCERT. I am just simplifying those lines and decoding in the simple words. So just try to understand the thing because I am not going out of the NCERT, but yes, sometimes it is very essential to take those parts of the NCERT. For decoding the NCERT, I am just venting out and taking that another material, searching for you for your easiness, right? So please cooperate with us. And whatever I am giving you here, it is again very important, and try to understand each and everything, whatever I am going to teach you in this particular class. Okay, so let us start a video. So let's just start it. You can see on the screen, that is the so starting from the introduction, nucleic acid. That is given in your NCERT, first line, first para. Nucleic acid are the two types. First is the DNA, another is the RNA. DNA, that is deoxyribonucleic acid, and RNA, ribose nucleic acid, right? So Friedrich Miescher, who discovered the nucleic acid in your DNA in 1869, and he called them nuclein, right? So due to the technical limitation, he failed to isolate the DNA intact, right? So DNA is the one type of the long molecular. DNA is the genetic material in most of the organisms. And DNA, if you see the RNA, so RNA can also act as a genetic material, but in some viruses like tobacco mosaic virus, HIV virus, retrovirus, poliovirus, etc. Rotavirus also, right? RNA is a more diverse than the DNA because RNA has the complexity like the mRNA, tRNA, rRNA, but DNA don't have those type of complexity. So functional diversity of the RNA is more than the DNA. Clear? Okay. Come to the next one point. There are three basic types of the RNA, that is mRNA, rRNA, and the tRNA. RNA is mostly function as the mRNA, that is the messenger, and next the structure of the ribosome, that is the rRNA, and last is the adapter molecule, that is the tRNA, which is the smallest one. So the RNA that function as the enzyme, which is known as the ribozyme. So that is acting like the enzymatic or the catalyst. Next is the DNA. That is the structure of the DNA. DNA is the long polymer of the 2' deoxyribonucleotide, and RNA is the long primer of the ribose nucleotide. Okay, one is the deoxyribo, another is the ribose. That is one is the difference in the sugar components. Both DNA and RNA are the polymer of the nucleotide, but the length of the DNA is depending upon the number of the nucleotides. Like one pair of the nucleotide having the one base pair. So in one turnover, they have the 10 base pairs, right? Next example is given that alpha into 174 bacteriophage having 5,386 nucleotides, and the alpha into 174 bacteriophage has a single standard DNA. So you have to learn that alpha 174 bacteriophage have the DNA, not RNA. Clear? That is the single standard, not double. Lambda phase or the lambda bacteriophage has 48,502 base pairs. E. coli bacteria has the 4.6 into 10 to power 6 base pair in its genome. Human beings have 3.3 into 10 to power 9 base pair in a genome. Clear? That is only the data which is given in your NCERT, and you have to learn data means as it is, whatever given. Next, if we're talking about the nucleoside and nucleotide, so first you have to clear your concept of what are the nucleotide and the side. So in case of the nucleoside, they are made up of the nitrogen base and the pentose sugar. In case of the nucleotide, they are made up of the nitrogen base, pentose sugar and the phosphate, right? So they are known as the building blocks of the DNA. Where DNA is the negative in charge because they are the acidic in nature and the presence of the acid, right? And another, if you're talking about histone, they are the basic in nature, and that's why there is a plus and minus when come together. It are not going to repel themselves; they just attract them, right? So adenosine, guanosine, thymidine, uridine, cytidine, all are the nucleosides. And if we say the nucleotide, so adenilic acid, thymidilic acid, guanilic acid, uridylic acid and cytidilic acid are the nucleotides. Nucleic acid, that is the RNA or the DNA, both are consist of the nucleotide only. Where the DNA and RNA both function as the genetic material, right? Whatever I am saying just before now, that DNA is present in the as a genetic material in the most of the organism, and RNA is less, like the viruses, TMV, rotavirus, and all. The structure of the polynucleotide chain. So, if we see the structure of polynucleotide chain, it is made up of the nucleotide as usual, pentose sugar, nitrogen base, and the phosphate, right? So, if you see the double standard model of the DNA, which is given by the that Crick and Watson. So, we have the two types of the nitrogen base. One is the purine, another is the pyrimidine. Purine and the pyrimidine, these two are the bases, right? In case of the purine, we having the adenine and the guanine. In case of the pyrimidine, we have the cytosine, thymine and the uracil. Uracil in case of the RNA. So here I want to explain you one more thing, that is the Charles-Graph rule. According to the Charles-Graph rule, the adenine is always paired with the thymine, guanine is always paired with the cytosine. Where adenine, if it is going to bind with the thymine, it is always with a double bond, and the C triple bond G. That means cytosine with the help of triple bond S, they are connected with the G, guanine. But Charles-Graph rule is not going to follow, or it is fail in case of the RNA because they have the uracil. Okay? How that chain is going to form, you can see here. That is the 5 to 3 prime in. What is meaning of 5 to 3 prime in? We will see later. So in case of the 5, here you can see on the 5 number of the carbon, the phosphate bond is there, and here the OH group. These two things is free actually, right? These two things is free. So anything can come and join in this area. Clear? So in the case of the 5 prime in, phosphate is present, that you have to remember, and in free OH group is present. So this is your ribose sugar. Here, this one is your ribose sugar. If it is the DNA, so we have the deoxyribose sugar it is. Here, this is all the deoxyribose sugar. And on base number 1, where the carbon is attached, that carbon is taking the 1-phosphate, and here also OH group is present, right? So from 1, 2, 3, 4, 5. On the 5 number, they are going to join with the all type of the nitrogen base, that is ATGC. Clear? Because we have the two bond here, one bond is this and another is this. So your backbone of the DNA is made up of the phosphodiester bond. Clear? So let's see what is meaning of the 5 to 3 prime direction, 5 prime to 3 prime in. So the new DNA strand need to form in the 5 prime to 3 prime direction. If the new DNA is going to form right? If any new DNA strand needs to replicate itself, it is always from the 5 to 3 direction. Clear? Leading strand and logging strand, two types of strand we have. In case of the leading strand, it forms the continuous because it is forming the 5 to 3 prime end direction. In lagging strand, it is the short segment, that is the Okazaki segment, that can also form the 5 to 3 direction, right? Come to the next, that is the salient features of the double helical DNA, which is actually given by the Watson and Crick. So it is made up of the two polypeptide chain, where the backbone constituted by the sugar phosphate and the base projected inside with the help of a hydrogen bond double and triple. The two chain have the anti-parallel polarity, means from the 5 to 3, another chain having the 3 to 5 direction. So polarity is slightly opposite, right? So it means if one chain is having the polarity like the 5 to 3 prime end, so another having the 3 to 5 prime end. The bases in the two strands are the pair to the hydrogen bond, that is the S bond, forming the bases or the base pair. Adenine form the two hydrogen bond with the thymine and opposite the cytosine with the 3 hydrogen bond with the guanine, right? So as a result, always purine from opposite to the pyrimidine. Purine from opposite to the pyrimidine. So this generates the approximately uniform distance between the two strands of the helix. The two chains are coiled in the right-handed, not left. It is the right-handed. So we having the V type of the DNA which is rotated on the right-handed side. So the pitch of the helix is 3.4 in this front, 34 in this front, and in the nanometer that is the 3.4 nanometer. And in case of the meter we have the 10 to power 9 meter. So there are roughly 10 base pairs are present in each turnover. So this is the picture you can see. Pyrimidine is the single cycle. Cytosine, thymine, uracil, that is the part of the pyrimidine. Purine, that is the double cycle, that is adenine and the guanine. Clear? And this is again, I am just showing you that minor and major group of the DNA. So this one is the minor. Here you can see this one is the minor and this one is the major part. Clear? So this is all base pairs, sugar phosphate, backbone. This is all the backbone. Okay? This is all the backbone. The last point is what? Okay. 10 base pair is in turnover. So we have, that is the turnover, right? And every turnover having the 10 base pair. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Okay? Just count it. Just assume it. Here. Come to the next, how the DNA is going to packaging. So the packaging of the DNA. Distance between the two base pair is equal to the 3.4 angstrom or 0.3 nanometer, and 1 nanometer is 10 to power 9 and 1 angstrom is 10 to power 10. So the total base pair in the human being, if we say the diploid cell, is equal to the 2 into 3.3 into 10 to power 9 base pair. So the typical mammalian DNA having the 6.6 into 10 to power 9 base pair. So how we identify or calculate it? Total length of the DNA is equal to the total base pair into the distance between the two base pairs. Clear? So in human being, the total length of the DNA is 6.6 into 10 to power 9 base pair, and the distance between the two base pairs is the 0.34 into 10 to power 9. If we cut it, what we got? 2.224 meter DNA. Okay? So the diameter of the nucleus is 10 to the power minus 6, or we can say 6 μm, and the size of the nucleus is 1.6. That is the perimeter. That is 1.7566 into 10 to the power 15 meter. So here you can see that sugar phosphate 5. Okay, that polarity. So you can see the polarity of this one strand. Let me show you. That is the polarity of this one strand. You can see. That is the 5 to 3 prime, right? And opposite, they having another polarity, opposite polarity, that is the 3 prime to 5 prime. Clear? So the distance of that sheet, you can see the 2 nanometer here, and distance between that two base pair is the 0.34 nanometer, and the turnover that you can see that first and second, and between that they have the 3.4 nanometer. And this is what the thing is given, how it is going to form. Come to the next one point, that how such a long DNA, that is how such 2.24 meter, 2.24 meter DNA, how it is going to pack in such a small nucleus, which is actually present inside the cell. That is the shocking point and very the magical point, right? So let's see the packaging of DNA in the prokaryotes. So prokaryotes don't have the nucleus, so they no need any type of the packaging. So in prokaryote, the negatively charged DNA is held with the positively charged basic protein, which is known as the histone protein. It is present in the non-histone protein, because they don't have the histone protein. Instead of the histone, they have the polyamide, which is present in the nucleide region. So DNA is present in form of the loop, right? In form of the loop, and their loops are held together by the basic polyamines. So they don't have the histone protein. Instead of histone, they have the polyamide. Okay? Let's see the difference between the heterochromatin and the monochromatin. So heterochromatin, actually where it is present, I am showing you. This is your nucleus, right? And in the nucleus we have the nucleosome, and somewhere we have in this part that darkest stain or the lightest stain things are present. So this one and this one. So let's take first heterochromatin, which is the tightly packed and stained darkly, which is called the heterochromatin, and it is the transcriptionally inactive. So this when you can take the heterochromatin. Okay. This is the euchromatin. So the chromatin is loosely packed. They are the lightest stain, which is called the euchromatin, and they are the transcriptionally active. Here. So come to next point. Difference between the histone and non-histone protein. So before taking their practicing in eukaryotes, we have to understand these terms first. The non-histone protein and the histone protein. So first, in case of the histone protein, they are a family of the basic protein, right, which associate with the DNA in the nucleus and condensing it into the chromatin. In case of the non-histone protein, so the protein which remain after the histone have been removed. Histone protein is having the 5 more types, that is the H1, or we can say H5, H2A, H2B, H3, H4. In case of the non-histone protein, they are the scaffold protein in a heterochromatin protein like the L-polycomb and the DNA polymerase. In case of the histone protein, they are the highly converged type of the protein across the species present, right? They are present in eukaryotes, and they help in the packaging of DNA into the nucleosome. In the another side, the non-histone protein, they are the less converged across the species, because they are always present in the prokaryote, and they play a role in the function related to the DNA. Let's see how the packaging of DNA in eukaryotes taking place in the next class, not now. So see you in the next class. Till then, just read your NCERT again and again, because things is from the NCERT only. So see you in the next video. Thank you so much for watching.